A key objective in microbiology laboratories is the identification of microbes. You have been utilizing the microscope and staining techniques to evaluate bacteria from different genera. Microscopy and staining, however, have their limitations in attempting to identify bacteria to the level of species. Let's consider Staphylococcus aureus and Staphylococcus epidermidis as examples. If we were to gram stain these two species, we would find that both are gram-positive cocci that grow in clusters. If we were then to acid-fast stain these two organisms, we would find that both are non-acid fast. So with each of the differential staining techniques, they appear identical, making it difficult to differentiate the two species. To aid in identification of bacteria to the species level, we must employ other tools in the lab. Biochemical tests, also referred to as differential tests, are important tests that allow us to evaluate differences in metabolic processes between species. While two species may look very similar under the microscope, they may be very different metabolically. We will be evaluating one type of biochemical test in the next lab session. You will be observing a carbohydrate fermentation test. Let's explore what carbohydrate fermentation tests are and what information they provide us with. Carbohydrate fermentation in bacterial cells results in the formation of an organic acid, or in some cases, an organic acid and a gas. Bacterial cells vary in the types of carbohydrates they are capable of fermenting. This is dependent on what type of enzymes exist in their cells. So, since we know bacterial cells differ in the carbohydrates they can utilize, we can set up a test in which specific carbohydrates are present in different test tubes to determine which carbohydrates are utilized. In order to determine whether the carbohydrate is being fermented, we must be able to detect a change in pH of the medium as the acid byproduct is produced. So carbohydrate fermentation media always contain a pH indicator that changes color as the pH changes. The carbohydrate fermentation media we are using in the lab is known as phenol red broth. Phenol red broth contains peptones, phenol red, and one carbohydrate. Peptones serve as a source of amino acids, and the phenol red acts as a pH indicator. Each test tube will have a different sugar. A small tube known as a Durham tube is placed inside the test tube in an inverted position. It is placed so that no gas is trapped in the tube prior to inoculation with bacteria. The Durham tube serves to trap gases if the bacteria produces gas as a byproduct of fermentation. The phenol red pH indicator will appear yellow at pHs less than 6.8, pink to purple at pHs greater than 7.4, and red in between those two values. The medium is adjusted to pH 7.3 when the broth is prepared so that it appears red. In order to conduct a carbohydrate fermentation test, the bacteria of interest are introduced using aseptic technique into phenol red broths containing various sugars. The broth is then incubated to allow growth and evaluated at approximately 18 hours. Broths older than 24 hours generally will not produce accurate results. The reason for this is that a bacterium capable of fermenting a sugar will first ferment the sugar as a source of energy. In doing so, the bacteria will produce acids that lower the pH, causing the phenol red to turn yellow. If the bacteria exhausts all the sugar through fermentation, it will then begin to metabolize the peptones that are present in the media. As the bacteria deaminate the amino acids from the peptones, they produce ammonia, which is basic. As the production of ammonia increases, the media then becomes alkaline and the phenol red changes to a pink or purple. At this point, we would not be able to determine if the bacteria fermented the sugar and then reverted to amino acids as a source of energy, or if it was unable to ferment the sugar and solely used amino acids as a source of energy. Let's consider how one would interpret the results of the phenol red carbohydrate fermentation test. If a bacteria is capable of fermenting the sugar present in the medium, it will produce acid as a byproduct of the chemical reaction. In doing so, the pH of the medium will be lowered. As the pH drops below 6.8, the phenol red indicator changes to yellow. After incubation, the presence of a yellow medium indicates a positive reaction, meaning that the bacteria fermented the sugar that was present. If the medium remains red, it is a negative result indicating that bacteria could not ferment the provided sugar. Some bacteria will produce gas in addition to the acid. The gas is detected using the inverted Durham tube. You must look to the top of the Durham tube to evaluate whether or not gas was produced. If gas was produced, there will be a bubble in the top of the Durham tube, indicating a positive result for gas production. If there is no bubble present, the result is negative, indicating no gas production. The image seen on the screen shows potential results from a phenol red broth carbohydrate fermentation test. Test tube C is the control. It has not been inoculated with bacteria. It shows you what a phenol red broth looks like when we begin this differential test. Test tubes A and B are both positive for acid production, as indicated by the change in color of the medium from red to yellow. Test tube A is also positive for gas production. The arrow indicates where the bubble in the Durham tube is located. Test tube D has become more pink in color, indicating that the bacteria have produced alkaline byproducts from peptone breakdown. And test tube E has been inoculated but shows no change. So let's check your understanding. The same organism is incubated in each of the test tubes seen on the screen. Can you determine what the results mean? I'll give you a moment. The results indicate that the test tube containing glucose shows a positive acid production with a negative gas production. The tubes containing maltose and sucrose are negative for acid production and gas production. So these results tell us that the organism can only metabolize glucose out of these three sugars, and that when it does so, it only produces an organic acid as its byproduct. Please look for the handout that accompanies this activity in your pre-lab assignment. It will be useful to have with you in the lab session.